case, we will start uh, from a diagram. There we will try to understand how we can divide this, and more importantly, you should know what type of changes which would occur here in case of cell cycle. So let's start with it. We have cycle because it's a cycle. So cycle is always round when, because it has no start and no end.
Yes, sir.
That's why it's a cycle. It is continuous process. We always divide these phases just in order to understand them. There is nothing like any other thing. Like it's a continuous process. We always divide it. Divide just for the sake that we can understand it, and we can elaborate this cycle, that what's going on at every single stage.
Okay, sir.
So, for instance, you have a cycle like this, and I can divide this into G2 phase. You can also like this. You have a S phase here, and then you would have a G1 phase. That's a little bit uh, larger in size. So this is G1 phase, this is S phase, this is G2 phase, and you would have mitosis phase, your mitotic phase. All right.
Yes, sir.
So this mitosis is actually the nuclear division. Now, what we are going to discuss shortly. First, we should know that what's going on here. Then we will talk about in detail about this. For instance, you are talking about the G1 phase, right? So what's going on in G1 phase? Remember, it is interphase. Interphase,
Yes, sir.
right? Are you getting?
Yes, sir.
Interphase. And at this point, you would have DNA duplication. DNA duplication. All right.
Yes, sir.
So another thing that you can add here, that is doubling of cellular content. Why? Cellular content. Because it's a stage which is going to prepare the cell for division. Because there would be, after this, there would be a mitotic phase where one cell will be going to divide into two different cells, right? So every cell has different material in it, and I mean there would be a nucleus, there would be a mitochondria, there would be Golgi complex, all other subcellular structures that must be present in a single cell. So in both of these cells, which will be divided from this main cell, the first thing is this cell has to divide its own content first. Are you getting this point?
Yes,
And
sir.
this would be at this point. It is very much critical phase. And if this stage will not carried out, I mean, it will not be here in this whole process. So definitely the cell is not going to divide. Are you
Okay.
getting this point? Is it? Do you want to say anything?
No, sir.
Ji beta. Okay, so at this point, another event that would be very much critical, that is synthesis of protein and RNA. Remember, what do you mean by this? Definitely, there will be some proteins there. There will be some DNA, RNA. All this is the content of the cell, right?
Yes.
So when I say that some protein is going to divide, what do you mean by this? Definitely, there will be some proteins, like in the form of enzymes. They may be structural proteins. More importantly, cytoskeleton and some other things. So all these are going to divide here.
Okay.
Duplicate, but I'm using the word here is duplicate. Everything has to replicate itself,
Yes.
right? In a very similar and identical form. Uh, you should keep in mind. So then you have the S phase, and from this S phase, it is what we call the synthesis phase,
Okay, sir.
right? It is
Thank you.
synthesis phase. And at this point, uh, the important thing that would be here, that is the replication of DNA.
Okay, sir.
Replication of DNA. And do you have any concept about the replication of DNA? What do you mean by this?
Sir,
What type of replication that always occurred in case of DNA? Replication of DNA. Ji beta. Ah,
translation,
what do you
transcription.
have? You replication, translation, and transcription, they are different concepts. But we are talking about right now, that is replication. Replication means that DNA, which is going to involved in this process that has to replicate. And I hope you should know about this semi-conservative replication. Semi-conservative
Yes, sir.
replication. It's a very different topic. Definitely, it's not the part of this discussion, but I just want to make you recall that this is a semi-conservative
Yes.
replication, and in this case, uh, let me tell
Strand,
you one thing. For
strand
instance, you have, oh yes, good.
bonds
So
to
there is a double. Hmm.
और उनके
All right.
ऑपोजिट वो जो होती हैं उनकी
There
जो
would be a template strand. There would be a. Hmm. Okay.
टेम्पलेट स्ट्रैंड होगा और एक मेन स्ट्रैंड होगा, इनकी बॉन्डिंग टूटेगी, फिर
Yes.
पर इनके वो
Exactly. That is what we call helicase enzyme will be there that will disunite or disintegrate or to divide the stand both of the strands.
यस सर.
All right, is it? And then at one strand, the process will be that will be repeated here to add the nucleotides. And according to semi-conservative replication, that you have the two strands on one side, this strand will be a non-template, another one is a template strand that will synthesize the complementary strand,
Yes,
the
sir.
very opposite. I mean, for instance, you have the thymine on one side, on the other side, what you would have? Adenine, is it? If you have cytosine, then you have a guanine. If you have adenine, and you have thymine. So this is how this is complementary combination.
Yes, sir.
Are you getting?
Yes, sir.
So, but hopefully in coming topics, we will talk about this, that about central dogma and specifically what is replication, what is translation, transcription, all these topics we will discuss in future, right?
Okay, sir.
Are you getting this point? Is it? Okay, let's move to the next one. So next thing that we are talking about, okay, okay, census phase. Then we have the G2 phase. What's about G2 phase? What
ग्रोथ
are
फेज,
the major changes?
ये इसमें एरर्स वगैरह फिक्स होते हैं
Uh, exactly. Errors and repairs. This is the one thing.
और
Though it will be fixed, all
माइटोसिस
those errors which are okay. Now before the one thing more here. That is the synthesis of tubulin protein from the spindle fiber. Synthesis of tubulin protein. One thing you have told me that error fixation.
Yes, sir.
Is it? The other thing you have the tubulin.
Okay, sir.
Are you getting this point?
Yes, sir.
So these are two main things, main events which would be changed here at this level, right? Are you getting this point? Is it? Okay. Now, this was the overall an idea about this, that what type of changes which are occurred here in case of cell cycle. The first one is G1 phase, where you would have the DNA duplication, or like all the content will be doubled. And then you have the synthesis phase. In case of synthesis phase, there will be the replication of DNA. This is the more important event here.
Yes,
right?
sir.
Then you have the G2 phase. In case of G2 phase, you would have the error fixation and the synthesis of tubulin proteins. What would be the function of this tubulin protein? I mean, is it going to form a spindle fibers, or it would be involved in mitosis? So
Yes, sir.
yes, it is. It is synthesized here at this level.
Yes, sir. Yes, sir.
Are you getting? Okay,
Yes.
fine. So now another thing that should that should be in your mind, that is G0 phase. G0 phase. Sometimes they may ask you in your MCQs that what is G0 phase.
Okay, sir.
And it is cell cycle arrest. I'm using the word here, arrest. Cell cycle arrest. What do you mean by this?
Failure?
There would be some cells in the body. No, not failure. I'm not talking about failure. There would be some cells in the body. They will stop replicating, or they will stop further dividing at certain point of their age, right? That is called cell cycle arrest. every cell in the body is not dividing every day. There are some cells which divide and reach up to the certain age, or they become mature, and then they stop dividing. And do you have an example for this?
Telomeres?
जी बेटा, बेटा
Okay, sir.
nerve cells. They are not. That's why I hope you should know about this. Once these are damaged in the brain of any person, so it is not possible to recover them as soon as. I mean, it takes long time, and even there are many neurons which are not able to further recover back or to get back to the original functioning. So it's not possible. Another thing that is red blood cells, RBCs. These are also the cells. Once they become mature, they have to go for death, right?
Okay.
So they will not renew themselves. Are you
Okay.
getting? So this is cell cycle arrest. What do you mean by the cell cycle arrest? It means that the cycle is going to stop now,
Okay, sir.
right? The cycle is going to stop now. It means that cell is not going to divide further.
Yes, sir.
Cell cycle continual mean those cells are going to divide in future. But once you are saying the cell cycle is at arrest, so it means there would be some cells which will not further divide. Are you getting this, Peter? Is it fine? Okay. Should we move?
Yes, sir.
Okay. So here's a mitosis. Peter, mitosis is nuclear division. Nuclear division,
Yes, sir.
and most of the time, this is the point that is the part of your extensive or structural question, that where you have to differentiate different events, like if you are talking about prophase, if you're talking about anaphase, I mean or metaphase or some telophase. So these phases you should know how to differentiate them, right, Buddha? Is it? So we can talk about one thing more in order to make this uh, more crystal clear. For instance, you have another thing. Uh, Wait a second. Here, for example, you have G1. Number
Yes.
of chromatids would be what? 1. At G1 phase.
Yes, sir.
What do you think? What would be after S phase?
2.
2. I'm talking about number of chromatids. Remember, number of chromatids. Right?
Yes, sir.
So, uh, G2 phase. What do you think?
2.
Remain 2. At prophase, what will be the situation? There are G
Uh,
at prophase
2
2.
young.
It's not going to change. Yes,
Yes.
exactly 2. And at metaphase,
Metaphase पे भी 2 ही होंगे.
yes 2. Then you have late anaphase. I'm not using the early anaphase here because we will discuss that in detail. So late anaphase. What do you think?
1 हो जाएगा.
It's, remember, cell is going to divide. Is it 1?
Yes, sir.
Because before that, in all other steps, the chromatid was duplicated.
Yes, sir.
In case of G1 phase, that's going to G2 phase. That was double in G2 phase as well, and prophase, metaphase, and that.
Okay,
It
sir.
means at telophase you would have only
1.
one chromatid. Right? This was the concept that you should know what's going on with the DNA during all this process. Right?
Yes, sir.
Is it? So you should consider human beings. Parent cell, how many chromosomes are there? 46.
Yes, sir.
G.
Yes, sir.
Number of chromosomes are
46.
46. Number of chromatids are number of chromatids I'm writing here. You should consider number of chromatids. Also 46.
Yes, sir.
Is there any change in it? So replication. Number of chromosomes during replication.
23.
G. In replication, it's going to replicate. Number of chromosomes. Do you think number of chromosomes will change in the cell when the DNA is going to replicate?
No,
Only
sir.
46, because that is a semi-conservative.
Yes,
That
sir.
is going to become. Oh, okay, fine. So number of chromatids would be already 46 in case of parent cell. So in replication, number of chromatids would be 92. Number of chromosomes are not going to change. They would be same. Number of chromosomes, whenever we say 46, it means that you have the type of, because in karyotyping, we always differentiate all these different types of chromosomes. So because after replication, you would have 92 chromatids, not chromosomes, number of chromatids. It means there would be a copy of that chromosome. And when we say the copy of that chromosome, we are not going to consider it's in number. That will always remain same. You
Okay.
will get this more clear when we continue this further. So let's talk about an interface again. I think it would be easy for you because we have already discussed. So just give a short glimpse to it. Interface. G. Interface. Okay, Raise, tell me.
Yes, sir.
Hmm.
डुप्लीकेशन होगी यहाँ पे सर जी.
Uh, it is interface. Is it?
G1 फेज ही नहीं था सर जी, इंटरफेस.
Definitely all different types of like G1 phase. Yes, in case of G1 phase, what type of changes? It is the longest phase. Remember, I'm talking about its characteristics.
ओके सर.
Longest phase. Is it?
यस सर.
Then next. DNA replication
यस सर.
in an in which part of this S phase,
एस फेज.
right?
यस सर.
Okay. Then any other change here? Uh, the chromatids which were first very condensed at this point of interphase, chromosomes are in the form of loosely coiled threads. We call them chromatin,
ओके सर.
chromatin, right?
यस सर.
Uh, centrioles have replicated in G1 phase of the interface. So it means centrioles are also going to divide here or replicate. So these are the changes which are commonly occurred in case of interface. You know what is the function of centrioles? Is it?
यस सर.
Okay. So now we are going towards mitosis. Mhm. I'm writing here. Mitosis. Uh, I think tab is not in a position, so I will not find it. So, mitosis. Let's start from mitosis. How many phases it has divided into? Four,
यस सर.
four phases,
यस सर.
right? So I think we are just here to revise all of these steps. It's very easy. First step is
प्रोफेज.
yes, but I wouldn't. Prophase. Okay, good. So tell me what the changes here.
सर
It is longest phase, remember,
प्रोफेज
in
में.
case of prophase.
यस सर.
Mhm.
वो न्यूक्लियर एनवेलप डिसअपीयर होता है न्यूक्लियोलस।
Chromosomes condensation
यस।
condensation of chromosomes at this point. Next.
न्यूक्लियस एनवेलप डिसअपीयर्स।
Ah, yes. Nuclear envelope disappears. Nuclear envelope disappears. Next.
न्यूक्लियोलस डिसअपीयर्स।
Okay, fine. That's also the part of it. Nuclear envelope. When nuclear envelope is going to disappear, it means that spot which was in the nuclear membrane, that is ultimately going to disrupt. Anything else that you can recall?
न्यूक्लियोलस डिसअपीयर्स।
In, uh, what do you think about? I mean, centrioles. What would be the change with centrioles?
सर जी
Don't
न्यूक्लियोलस
you
भी
think so? The centrioles, the centrioles will go. G, sorry.
न्यूक्लियोलस भी नहीं डिसअपीयर होगा?
Yes, you can add. You can say I said already that nucleus is ultimately is going to disappear because nuclear envelope is no more,
ओके सर।
right? So you can add here. You can add here, but that's not the major event here. That's the ultimate event because of the nuclear envelope. Because nuclear envelope is going to disappear now. So any other change
सर जो
about centrioles? Think about centrioles.
रोल्स
What would be the change here with centrioles?
सेंट्रियोल्स ऑपोजिट
Hmm.
पोल्स की तरफ मूव करना शुरू करेंगे मेरे ख्याल में।
Yes, opposite movement, opposite pole. You can write it about centrioles.
यस सर।
Okay. Anything else? Jeevan, anything else yes, you have told me that disappear, the nuclear envelope is disappeared, that start is going to be, I mean, so these are all the changes which are actually happen in case of prophase,
यस सर।
right? All right.
यस सर।
Now then you have metaphase.
यस सर।
Think about metaphase.
सर मेटाफेज में क्रोमोसोम्स विल लाइन अप एट द इक्वेटर।
Yes, line up at equator. Here, we have five. Yeah.
सर आपकी आवाज़ फिर से नहीं क्लियर आ रही है।
Oh, okay, okay. ये मैं it's okay. Now it's clear. Now it's clear.
यस सर।
Okay. So tell me about the metaphase. Metaphase.
सर क्रोमोसोम्स विल लाइन अप एट द इक्वेटर।
Metaphase. Yes, good. Good. Metaphase is going here alignment. Alignment of chromosomes. On what? Where?
ऑन द इक्वेटर।
Go in deep, yaar. Go in deep. Huh?
ऑन द
Yes,
इक्वेटर।
that's form that's equator, but think about it more that for instance you have this shape of chromosome here.
यस।
So what is this? What is this point on which I am just
सेंट्रोसोम।
filling this space? What is this? Sorry, it is.
सेंट्रोसोम।
Is it centrosome? Centromere. Is it
सेंट्रोमेयर सॉरी।
centromere?
सेंट्रोमेयर यस सर।
Yes, sir. Okay, no issue. Centromere. Anything else? Is there any specific protein here by which the centromere is formed? Do you know or do you remember the name of it? Kinetochore? Is there any
यस सर।
kinetochore? Kinetochore. This is the specific protein by which this centromere is formed. So this kinetochore is the point on which there would be the spindle fibers which will come to attach here,
यस सर।
right?
यस
They will
सर।
come to attach here, and this is how uh, definitely on the other side you have the centrioles, which was the part of centrosome first,
यस सर।
right? So this kinetochore is the point at which the spindle fibers from the opposite pole of the cell
will attach.
They will become to attach on it, right?
Yes, sir.
So that's why it will be aligned on the equator. This is the whole concept. And whatever the number of chromosomes there is no replication, no duplication. It doesn't matter the how many number of chromosomes there. They all will align in a very similar pattern, right?
Yes, sir.
Are you getting? Then you have anaphase. Come to the anaphase. Let's complete it as possible.
एनाफेज में सर जी जो स्पिंडल
जी
फाइबर्स हैं
यहाँ
शॉर्टन हो जाएंगे.
जी सॉरी
एनाफेज में सर
anaphase.
स्पिंडल फाइबर्स जो हैं वो शॉर्टन हो जाएंगे
Okay.
एंड दे
Anaphase.
विल
Here is
पुल द.
anaphase. जी बेटा. Sorry. Anaphase. What's going on? What's going on at anaphase? Rapid stage of mitosis. It's a rapid stage of mitosis. The centromere split into two and spindle fiber pulls. Two and spindle fiber pulls the daughter chromatids towards. What is the key change here at anaphase?
सर
जी
स्पिंडल
बेटा.
फाइबर शॉर्टन हो
Yes.
जाएंगे और वो.
Key change. Yes. Actually, splitting होगी. Splitting is the key point here.
यस सर.
For instance, there's a pair of like this. Here you have on the kinetochore, it is attached like this. In anaphase, you would have this condition.
यस सर.
This is a splitting, and this splitting is occurred in case of anaphase.
यस
Is
सर.
it fine, Beta? But remember, you have to draw a complete diagram. For instance, you have taken 4 chromosomes. You have to justify at the end of this that what uh, happens with those 4 chromosomes by showing them in a very proper cells, right? Their splitting, their alignment, all these things should be very streamlined. So I'm just here to tell you what are the key changes which are occurred here, right?
यस सर.
So when you would have the diagrams there, so you can easily differentiate that is it anaphase, is it telophase, is it prophase, late prophase, early prophase. So you should be able to differentiate all this.
यस सर.
Okay, beta. So anything else? Telophase. One thing about telophase.
टेलोफेज में सर न्यूक्लियर
Key
एनवेलप.
change that
reappearance of nuclear
oh,
nuclear
yes,
envelope.
it is very opposite to the prophase.
Yes, sir.
Whatever changes occur in case of prophase, all changes will be reversed in
Yes.
case of telophase. Right? You said chromatids reach pole of the cell.
Yes, sir.
Definitely all the chromosomes will reach to the opposite poles.
Yes, sir.
Got it?
Yes, sir.
Then you have the spindle fibers. They will disintegrate because they are no more going to use here now because their function is completed. So those fibers will be disintegrated. Then
Yes.
you have nuclear envelope that is reformed and nucleoli reappear. What nucleoli? That nucleolus,
Yes,
which
sir.
was disappeared previously in case of prophase, that is going to reverse. Right, beta?
Yes,
So
sir.
what is the next step? Hurry up. What is the next step after this?
Sir,
Just
this
all
centrosomes.
cell division.
Yes, sir.
Cell is completely prepared to divide now,
Yes, sir.
and that is cytokinesis. Cytokinesis.
Kinesis.
Cytokinesis. Are you getting this? Cytokinesis. Are you getting?
Yes, yes.
What's going on in cytokinesis? Cytokinesis is the division of what?
You know of this.
Division of what?
Sir, वो cell
Cytoplasm.
cytoplasm divide हो जाएगा. Yes, sir.
That will convert into how many cells?
2 cells.
2 cells. 2 daughter cells. What we call them?
Daughter cells.
Geodes. 2
Daughter
Two.
cells.
daughter cells. And which leads into G1 phase of the interphase?
Yes,
They
sir.
will go into the interface. Definitely they will get grow and I mean uh, start duplicating or increasing the content of the cell, and the cell organelle is evenly distributed towards the two poles of the telophase. But how this cytokinesis will take place? There would be a pinching of the cell membrane in the middle,
Okay,
right,
sir.
towards inside, and in plant cell, new cell wall is formed. This is the key difference that always uh, mostly uh, changes which take place in this case. All right.
Sir.
So I didn't draw a diagrams here, but it is your assignment that you should draw a diagram of uh, I mean prophase, metaphase, anaphase, telophase, and cytokinesis.
Yes,
And
sir.
I want to have that should be in your. I mean, if you don't want to draw, so you can pick different diagrams from somewhere and write about them, that what type of changes which occur there,
Okay, sir.
right? So at least you can practice on it.
Okay.
So you don't need to remember anything specifically, but you should know the changes.
Okay.
Changes are important, right?
Okay.
So the next point is that we will discuss in our next lecture, that would be significance of mitosis.
Okay, sir.
That is
Significance of.
significance of mitosis, because from here you will understand then, because it is remember, it is cell cycle in which you are talking about mitosis. What is the key point in mitosis and in meiosis that we will discuss in our coming lectures? Give it up.
Sir, जी, division
Number
of.
of chromosomes will remain same. Number of chromosome will remain same in case of prophase, or in case of what, mitosis. So that's all for today, beta. Okay, fine. I hope we will. I don't know. Tomorrow I will not be available here because I will travel, and
Yes, sir.
but when I will reach there. So on Sunday, if you will be available, so I will try to connect with you. Okay.
Okay, sir. जी, okay.
Otherwise, you have the assignment that you have to send me all the videos specifically with date, that on what date we have studied for.
Yes, sir.
Right?
Okay.
Okay, Race. Thank you very much.
Okay, sir.
God bless.
No, worries.